unseen enemy, we use and have benefit of unseen friend. Only with some knowledge and techniques we can feel its presence or absence. Koch discovered TB and Pasteur discovered pasteurization. It was only in 1983 that the HIV virus was isolated and identified by a researcher at Pasteur Institute in Paris, in France. 3 Nobel laureates in 2020 win the Nobel Prize for the discovery of hepatitis C. This is a concise course about bacteriology. I am Dr. Taha and I take you in a journey, a short journey. We learn, we enjoy. Let's start. Introduction. Microbiology, it's 3 syllables. Micro in Latin means small, logos means science, bios means life, and it's the science dealing with these minute microorganisms unseen with naked eyes. Why microbiology? Why we study this subject? Microbiology really it's extensive science, and it's in every side in our life. We find it's we can divide it into bacteriology, virology, mycology, protozoology, and we can say it's clinical microbiology. When we deal with patients and the hospitals, we can say environmental microbiology. When we deal with sea or soil, we can say industrial microbiology. When we speak about materials, we can say food microbiology. When we take samples from food and count the bacteria, and the total bacterial count is more than, less than, and we evaluate this food. You may have the chance to save a human life. If you study microbiology, you can invent a new product, or it changes the method of thinking of people. Really, it's interesting science. Microbiology, no doubt that microorganisms exist on the earth, water, ground, air, millions of years before man. The life of ruminant can't begin or continue without the rule of ruminant microflora, which is bacteria, or microfauna, which is protozoa. In fact, the relationship between man and the microorganisms start as early as beginning of his existence on the earth. He observed the putrefaction of carcasses of animal birds and himself. Putrefaction, the process of decay or rotting in body or other organic matter. Ancient Egyptians could stop putrefaction of carcasses of their kings and preserve their holy mummies for several thousands of years by famous Egyptian art and science of embalming, by adding known and unknown chemical substances which kill the body. No, the real microbial cause. He noticed sugar fermentation, made alcoholic beverage. He experienced several bacterial, viral, parasitic diseases. He tried herbs and magic. He made bread. He made cheese. He made finger. Man tried to understand the usual changes resulting from microbial action as purely chemical process. Life itself, when seen in a small creature like worms and insects, was explained by spontaneous generation. Aristotle, the life is a chemical interaction between organic matter, water, air, and heat. Only in 1668, ready, an Italian scientist doubted in the theory and in simple experiment could prove that maggots are not spontaneous creatures from dead matter, but they were the hatching larvae from eggs laid down by flies on piece of meat. Maggot fly larvae would appear on the uncovered meat but not on the meat in the sealed jar. In his second experience, Riley used gauze to cover half the jar to allow air but not flies. Only in 17th century, two major achievements regarding the discovery of microscope. Robert Hooke in 1665 discovered compound microscope. He was able to see individual cells of living creatures. Leeuwenhoek in 1773 with his single-lens microscope with magnification power 300x discovered the microbial world. He described and drew microorganisms for the first time. He published his discovery in Journal of the Royal Science of London in 1683. Pioneers or the fathers of the microbiology. Louis Pasteur discovered pasteurization. Robert Koch, Koch's postulate of the disease transmission. Joseph Lister discovered sterility. Koch taught us his postulate to prove that the causative agent to make the disease. He taught us the bacterial isolation in pure culture, discovered tuberculosis, anthrax, cholera, invent tuberculin test. He won Nobel Prize in 1905. He died in 1910. Physiology of bacteria. Types of bacteria according to source of energy. Chemotrophic bacteria require energy from chemical substance. Phototrophic bacteria require energy from sunlight. Types of bacteria according to source of carbon. Heterotrophic bacteria can't use only CO2 main source of carbon but require organic carbon. Autotrophic bacteria can utilize CO2 as main source of carbon. The family Bacilliaceae sporulation. Endospore formation in the family Bacilliaceae change typical vegetative form into spore forming sporogenesis, exhaustion of the material, nutrient material, dryness or change of pH, accumulation of toxic substances. The types of spore may be spherical or oval, may be central or peripheral, and it's a method for typing of Clostridial species. Methods for identifying unknown bacteria. Identification of unknown bacteria on one of the most important tasks of microbiologists. Morphological examination, shape, size, arrangement on the dish after cultivation, presence of capsule, spore, metachromatic granule, the characteristic of every bacteria, shape under dark field microscope, motility of microorganism, the microorganism either motile or non-motile, gram stain, the crystal violet, iodine, carbulfoxin, alcohol, safranin or carbulfoxin. The bacteria either gram positive or gram negative. The gram positive take the violet stain and resist decolorization by alcohol. Gram negative stain, gram negative bacteria, it take the crystal violet, after that decolorized by alcohol, and after that take the red color of the carbulfoxin. Zell-Nelson stain, the primary stain is carbulfoxin. The secondary stain is methylene blue, and it's invented by Zell and modified by Nelson, and before them Ehrlich suggests using of carbulfoxin, which is the Cranibacterium, take it and resist decolorization with acid alcohol. The acid fast bacteria appear pink, while the non-acid fast appear violet in the background. Colonial morphology in several bacterial culture on solid media, shape, size, color, margin, consistency. The growth on broth media, growth on blood agar. Biochemical identification depend on the presence of different enzymes, different bacteria, addition of substrate, different substrate, and indicate the presence of the product. This is the principle of the biochemical test. The enzyme either present or absent. The biochemical test: oxidase, catalase, coagulase, sugar fermentation test, litmus milk, nitrate, urease, gelatin, IEM, VIC, triple sugar iron agar, the animal inoculation, serological identification, phage typing, molecular